about the initiation step of TCA cycle, which is formation of acetyl CoA. TCA cycle is the second and the important pathway of cellular respiration. It is important because here we can see the generation of reducing equivalents, an amount of 3 NADH and 1 FADH molecule to supply for electron transport chain to make energy either in the form of chemical, that is ATP, or thermal energy, which is heat, to maintain the body temperature. In order to start the TCA cycle, we require two molecules. Those are acetyl CoA and oxaloacetate. Let's see the formation of acetyl CoA in a cell and what are the various sources of acetyl CoA in a cell. The citric acid cycle is the central metabolic hub of the cell. Why? The three different energy yielding molecules we consume via diet, which are amino acids, fatty acids and carbohydrates, are breaking down to the fuel molecule acetyl CoA in order to enter into the second stage of cellular respiration, that is citric acid cycle. Hence, citric acid cycle is considered to be central metabolic hub of the cell. Where does citric acid cycle takes place in eukaryotic cell? It takes place inside the mitochondria, in contrast to the glycolysis which takes place in the cytoplasm. The citric acid cycle is also known by different names. In the honor of the scientist who discovered citric acid cycle, it is called Krebs cycle, or it is called tricarboxylic acid cycle, as the first compound citric acid formed from the first chemical reaction of TCA cycle or citric acid cycle contains 3 carboxylic group. It is also called tricarboxylic acid cycle. What are the different types of chemical reactions takes place in citric acid cycle? Includes a series of oxidation reduction reactions to generate reducing equivalents NADH and FADH2. As we know, the hydride ion, which contains energy, is captured by these reducing equivalents in the oxidation and reduction reactions. And at the end of the TCA cycle, the regeneration of oxaloacetate is also very important step. So how does the citric acid containing 6 carbons lose 2 carbons obtained from acetyl group? Via decarboxylations happening in the TCA cycle, the citric acid 6 carbon compound will lose 2 carbons in decarboxylation reactions and finally converts back to the oxaloacetate, which is very important because in order to run future TCA cycle reactions, oxaloacetate is important compound to react with the acetyl CoA and start TCA cycle. This picture clearly depicts the different sources of acetyl CoA, which are fatty acids, pyruvate formed from glucose, amino acids, ketone bodies. During the starvation and difficulty periods of human body, the acetyl CoA is synthesized from fatty acids, amino acids and ketone bodies. During normal periods, that is after the intake of meal, it is obtained from pyruvate formed from glycolysis. Here the important fact to be understand is acetyl CoA is a 2 carbon compound which is formed from pyruvate, which is a 3 carbon compound. Pyruvate is forming from glucose, which is 6 carbon molecule. Hence, one molecule of 6 carbon glucose splits to 2 molecules of pyruvate. So from one glycolysis pathway we obtain 2 molecules of pyruvate. 2 molecules of pyruvate enters into the mitochondria with the help of pyruvate dehydrogenase converts into 2 molecules of acetyl CoA. The formation of acetyl CoA from pyruvate takes place in the mitochondria. As pyruvate has access inside the mitochondria via pyruvate shuttle present in the inner mitochondrial membrane, the pyruvate has access to mitochondrial matrix. So inside the mitochondrial matrix there is pyruvate dehydrogenase, which has 3 subunits carrying different types of reactions in order to make acetyl CoA to initiate TCA cycle. It has pyruvate dehydrogenase, dihydrolipoyl transacetylase and dihydrolipoyl dehydrogenase. So the pyruvate 3-carbon compound to convert to acetyl CoA 2 carbon, it has to lose 1 carbon in the form of carbon dioxide. Here we see the decarboxylation, release of carbon dioxide. And acetyl molecule should be attached to the coenzyme A group. So the coenzyme A is added to the acetyl group with the help of dihydrolipoyl transacetylase. At the end of this reaction, there is formation of NADH plus H plus. So we can conclude from each pyruvate formed from glycolysis, so there is one NADH plus H plus molecule generated within the pyruvate dehydrogenase complex. The important things to be noted further are cofactors of pyruvate dehydrogenase. TPP, lipoic acid and FAD, NAD, these are important cofactors of pyruvate dehydrogenase complex which are derived from the B complex vitamins. So in case of vitamin B complex deficiency, we can expect there is like speed of the TCA cycle will be slowed down, or the number of times the TCA cycle has to be taken place will be reduced due to the lack of vitamin B complex derived cofactors which are TPP, FAD, NAD, lipomide, etc. So that's why in case of vitamin B complex deficiency, there is weakness manifested in the patient due to the energy generation defect. As there is no TCA cycle taking place or less number of times TCA cycle is taking place, the energy yield, the energy formed in the cell in total in the human body is decreasing, so it affects the human energy levels which is manifested in the form of weakness.